Hi, everyone. Welcome to What's New in MPMI. We'll wait a few minutes while people are logging on. I see people coming in the door, the virtual door. Great to have all of you. While people are logging in, so first of all, welcome to What's New in MPMI. And while people are logging in, I love to see where's everybody from. So if you could type where you're from into the chat, that's fun for us to be able to follow and see where everybody's coming from. So Sridhar Ranganathan is coming from Chennai, India, Radin from Spain, Jordanis from Greece, M. Mohammadi from, I'm not sure where, Adrian from, oops, going a little fast now, Brian Push from Elma, New York, Pascal Raté from France, Adrian Gonzalez Ortega Vilazan from Spain, Devita Srivastra from India, Maria Reguera from Spain, from Madrid, so many people. Alefia from Houston, Kouakou from Cote d'Ivoire, Mojtaba from Palmetto, Bikash Raul from India, people from all around the world, Canada, Davis, Kansas, Peru, Sunil from India, Agoto Domonkos from Hungary. So very exciting to see everybody joining from around the world. This is the really one of the reasons we started this. IS-MPMI, the International Society of MPMI, is an international society. We live all over the world, all around the world. And one of the goals of What's New in MPMI was to increase connection and inclusion. So now that most people are on, I wanted to welcome you to What's New in MPMI. This is the virtual seminar of the MPMI Journal, where we highlight papers and authors from MPMI. So this was originally started to increase both inclusion and engagement. So it's a way for us to connect with all of you across the world. It's a way for you to connect with us, for you to connect with authors, for authors to connect with readers and listeners outside the pages of the journal. And you can actually ask them questions in real time, which is wonderful. And of course, you can always contact the authors by email, because when you go to the article, there's that little email you can click, and so you can contact them from there. So I was pretty excited because today is we're starting our 2023 series, and it's going to be great. Our next one is scheduled for March 7th with Thomas Griebel. I think that's going to be great. And we're trying to schedule another one for February, and more on that later. And Karen will type into the chat where you can go to register for upcoming talks, and also where you can find the recording of this later. It's going to be posted later. So since 2020, that's a long time. This is our 4th year doing this virtual seminar series, and it's been a lot of fun. And that is why I'm continuing to do it, even though I stepped down as editor-in-chief on December 31st. So no longer doing that, but I decided to stay on as host and organizer of What's New in MPMI, because I really like connecting with all of you around the world. I love hearing new science and just what's happening. Thanks for all the little hearts, by the way. That's kind of cute. Okay. One final thing is we're trying to increase accessibility, and that's why we have the closed captioning. I know it doesn't always perfectly capture scientific writing, and for that we're sorry. Also, personal names usually falls down on that. So if anybody would like to volunteer to help us edit the transcript afterwards, then we can provide an edited transcript to the talk on, to our Zoom talk on the recording, and that will help to improve accessibility for people who are hearing impaired, but also for people who, for whom English is not their first language, or people who just want to listen to it again and want to capture more of that info in the closed captioning. So please email me or type your name into the chat if you would like to volunteer to help out your colleagues in this way by editing the transcript. Okay, so Today's talk is by Emma Gachomo from the University of California, Riverside. I'm delighted to have her here. She published a very interesting paper on one of my favorite interactions, the rhizobium legume interaction, except with a nice little twist. Guess what? They don't just interact with legumes. And so that's the story we're going to be hearing today that she's going to be taking us on. So I'm going to turn this over to Emma to tell us about her work. At the end, we'll be answering questions. So please type your questions into the Q&A. We will not be answering questions from the chat, just the Q&A. And you can type them at any time and I will answer them at the end, read them off at the end. Okay, thanks. Emma, would you like to share your screen?
So good morning, good afternoon, good evening, wherever you're joining us from. Thank you very much for attending the talk. And Jeannie, thank you very much for the invitation. I'm happy to be here. So my talk is on Bradyrhizobium japonicum alters Arabidopsis thaliana root architecture through regulation of Auxin efflux transporters PAIN2, PAIN3, PAIN7, and ABCB19. As she introduced, I'm an assistant professor at the University of California, Riverside. So the question you might be asking, why did we choose to study this bacteria? Why did we choose to work on it? I had worked on it as a postdoc some years back, and we had gotten very interesting results, and therefore I wanted to come back to it. So briefly, I'll present also the results that we had obtained earlier. Bradyrhizobium japonicum, which I'll later just refer to as B. japonicum, is a soil-borne bacteria. And it was originally isolated from Cameroon, Africa, from cowpeas, and characterized by a research institute in France, in Montpellier. And then they, hence the name B. japonicum IRAT FA3, then labeled it strain number 3. It's a plant growth promoting rhizobacteria. And as with many plant growth promoting rhizobacteria, they are known to improve plant growth and also improve plant tolerance to biotic and abiotic stress. Now, the reason B. japonicum or the Bradyrhizobium genus is important is because it's very ubiquitous in the soil, and therefore it's likely to interact with many different plant species. And therefore, studying it is going to, we thought was going to give us a broad understanding of how plant growth promoting rhizobacteria in general interact with plants. The mechanisms through which plant growth promoting rhizobacteria interact with plants are very variable, and there are many of them that have been labeled out there, but it doesn't mean that every bacteria species you see is going to be interacting with the plant using the same mechanism. So they could be specific to a species, they could be specific to a genus. And therefore, we wanted also to understand the mechanism that B. japonicum was using to interact with plants. And some of the mechanisms that have been known include phytohormone production, nitrogen fixation, siderophore production, ACC deaminase production, and many others. And therefore, we embarked on a journey of studying this interaction of Bradyrhizobium japonicum with plant species. The study we did earlier, we had also included corn in the study, and we found that B. japonicum was increasing corn biomass. If you can look at the two plants here, two groups of plants, and also increased root biomass. We also observed that it increased the leaf surface area, both in Arabidopsis and in corn, meaning that it was not limited either to monocots or dicots. And this increase in root in leaf biomass was also significant, as you can see from our table here. And therefore, it was an interesting bacteria to study, in that whatever results we obtained in dicots or in monocots, whatever results we obtained in dicots could also be possibly translated to monocots. And we had also observed that it increased yield. And this study here, the data I'm showing here is for Arabidopsis. And as you can see in the graph, the significant increase, we noted it very early, this around 24 days after germination. if you're familiar with the Arabidopsis, this is just about the time that the seeds start forming. And with time, we noticed that the differences between the inoculated and uninoculated plants were growing bigger. And by about 38 days after germination, in the non-inoculated plants, we had about 30 seeds, 30 calyx. Calyx are the pods that contain seeds. So we had about 30 calyx in the control plants, where in the inoculated plants, we had about 200 calyx per plant. So you can tell that there was such a big difference and a very significant difference between the two. So this data we obtained from these plants that we had grown, and we had grown them just in soil that we had obtained from the fields. So it was just a regular soil that you would have, and it wasn't that nutrient rich. And you can tell that in the case where the nutrients were limited, the bacteria just did a very good job in enhancing the plant growth. And we didn't give anything extra to this group of plants. We just added the bacteria to it. And as you can see, it was not only increase in biomass, it was also increase in the yield, because the stalks are taller, therefore they're going to bear more, they're going to carry more seeds. Then we also wanted to know if the grain yield was increased. And for that, we measured, or we counted the number of seeds per calyx. And as you can see from this picture here, the calyx were not only longer, they also had more seeds. And we found that the average number of seeds per calyx were about 35 in the control, while in the PGPR inoculated plants, they had about 75. So there was this significant increase in the number of seeds per calyx. And this tells us that if you're looking for biomass yield, just leaves, stems, the PGPR can help plants in obtaining that. But also if you're looking for yield in form of grain, PGPR can also play a role in increasing grain. And with that, then we were curious to know the mechanisms behind this, because if it's increasing all this and doing all these good things, what mechanisms is the bacteria using in the interaction with plants? And therefore, we carried out this other study. And here we focused mainly on the root architecture. We wanted to see what it was doing below ground. And this study we carried out with Arabidopsis. And here we can see that when we inoculated the plants with the bacterium, the primary root was shorter in the inoculated plants. And the number of lateral roots increased. And we did do some measurements which showed that there was a significant reduction in the primary root, and there was also a significant increase in the number of lateral roots. And this significant increase started as early as 2 days post-inoculation. And by 8 days post-inoculation, there was a 70% increase in the number of lateral roots produced in the bacteria-inoculated plants, showing us this very robust increase in the number of roots. And this is just within 8 days. So you can imagine if it was a whole growing season for a plant, beyond which maybe could be months. So the effects could be larger than just what we are seeing here. Now, this phenotype in plants where we see reduced primary root length and increased number of lateral roots is something that's observed with application of Auxin, exogenous application of Auxin. And therefore, our questions were, one, was the bacteria producing Auxin itself and feeding it to the plant? Or was it inducing the plants to produce Auxin? The other thing that's important for lateral root production is Auxin transport. So was the bacteria interfering with Auxin transport? Because in lateral root production, there's an Auxin gradient that has to be created, and the Auxin concentration at the place where the primordia forms is usually high, meaning that the bacteria had to be interfering with the Auxin gradient in the plant. And now to confirm all these studies, then we carried out some studies. And the first thing we did was confirm what we had said. We had said that this phenotype looks similar to application of Auxin. So we did carry out a study where we applied exogenous Auxin to the plants and looked at the effects of that compared to the effect of the bacteria. And we found that when we applied auxin in form of NAA, which is, we used two analogs of auxin, NAA and IBA. When we applied NAA, there was that sudden increase within 2 days, that sudden increase in the number of lateral roots that were being formed, and they continued to increase over time and then leveled out. And then when we used IBA, the process was slower, but eventually also there was that increase in lateral roots over time, just within the time period that we're measuring. The bacteria was slightly slower, but it did also increase. It also did increase the number of lateral roots. We're seeing the bacteria here in the gray with stripes across. These were the ones that we inoculated with bacteria, and we found also that it was increasing the lateral roots also over time. Although it was slower, it did increase with time. And therefore, that effect was similar to what we were seeing with exogenous auxin application. These chemicals, the auxin analogs, NAA and IBA, were dissolved in DMSO, and we wanted to see if DMSO had an effect either on the lateral root production and also on the bacterial growth. And we, the black bars are where we have DMSO. And here we found that when you added DMSO just to the control, it did not have an effect on the, it did not seem to have any effect on lateral root production compared to the control. And also DMSO did not seem to affect the bacteria in production of lateral roots. And then the other question we wanted to answer is if addition of auxin was in any way influencing the number of bacterial cells in a culture. And we found that there was no significant difference between the control and addition of NAA in the number of CFUs per root per millimeter. So there was no significant effect of the exogenous auxin on the bacterial cells in the plant that were formed in the other root. They did not either increase them rapidly or decrease them rapidly. Then the other question, if you remember, that we had asked in the previous slide, was the bacteria producing auxin or was it inducing the plant to produce auxin? And to do that, we measured the concentration of auxin in the liquid culture that was we had grown the bacteria in. And we found that there was some amount of auxin in the culture. And then we supplied a precursor of auxin, tryptophan, which is a precursor of auxin in plants. And we supplied it in our growth media. And we found that there was a significant increase in the amount of auxin or IA that was produced when we added the precursor. And this increased with time. And you can notice that after 48 hours, it was the difference was even greater than it was at 24 hours, showing that the bacteria did produce auxin. And if you supplied a precursor for auxin, it had potential to increase the amount of auxin that was available. And then we also wanted to find out if the bacteria influence the auxin response in the plant. And with that, we measured expression of several genes that are involved in auxin within the plant. For example, GH3, which is an auxin responsive gene. And we found that it was increased as you added bacteria to the plant. GH3 is auxin responsive, and it's responsible for auxin conjugation, meaning that if you have too much auxin supplied to the plant, then GH3 is activated to convert that auxin into another form so that it kind of reduces the amount of auxin available inside the plant. And we found that it was increasing in the plant. Therefore, the bacteria was triggering an auxin responsive genes within the plant. Then we're also curious to see the amount of auxin within the root system. And for that, we used an auxin responsive promoter fused with a gas reporter gene. And in the control, we could see that auxin was limited to the root primordia, the lateral root tip, and also the tip of the root, the primary root, at the meristem. But then in the B. japonicum inoculated plants, we found that auxin was not only in the primordia, in the lateral root and the root tip, but it was also all over the plant, indicating that there was a high concentration of auxin in the plant when we added the bacteria to the root of the plant. And therefore, there was that, we had an inkling that the bacteria then was also responsible for auxin regulation, in the sense that we can, as you can see, these plants are the same age, these roots are the same age, but you can still see that the primordia is more advanced in the B. japonicum inoculated plants, and also the lateral root is already emerged, kind of fully emerged in the B. japonicum inoculated plants. So we thought that the bacteria could also be involved in auxin transport. And therefore, we decided to use auxin transport inhibitors. Now, for auxin transport in the plant, there's auxin transport into the plant, which is auxin influx, and then there's auxin transport out of the plant. And for our case here, we decided to work with auxin efflux, that is auxin being removed from the plant. And we used an auxin efflux inhibitor, NPA. And when we added it to the culture, we found that the number of lateral roots produced by B. japonicum reduced to the level of the control. Well, in the case of NAA, when we added the auxin efflux inhibitor, we found that it reduced even below the level that it had reduced for B. japonicum, and therefore showing that there were kind of some differences between using a living organism and just having the chemical available. And from our study here, we were able to see that addition of an auxin efflux inhibitor reduced the B. japonicum induced lateral root production, because when we just have B. japonicum, the number of lateral roots produced are at this level. But when you added the inhibitor, it reduced them significantly to about an average of 10, which was significant in our case. We also observed that B. japonicum reduced the primary root. No, adding NPA reduced the primary root length here, as you can see from this study. And therefore, this pointed us to efflux transporters in the plant, and we wanted to see if B. japonicum had any effect on the expression. And we did a qPCR analysis, as we can show here. Excuse me. And what we observed was that when we use the efflux transporters that are found in the roots, that is the pins and ABCBs, pin 1, pin 2, pin 3, and pin 7, and ABCB4 and ABCB19, we found there were differences in expression. Now, there are two groups of auxin efflux transporters. We have the pins and we have the ABCBs. The pins are responsible for the polar transport of auxin, while ABCBs are important for intercellular transport. And what we observed here is that pin 3, pin 7, and ABCB19 the expression increased when we added the bacteria to the soil, while pin 1, pin 2, and ABCB4 did not seem to be much affected. While ABCB4 did increase, but the increase was not significant. And from here, we wanted to see if there was any other effect that the bacteria was having on auxin related genes in the plant, other than just the efflux transporters. And we did an RNA-seq analysis. And what we found is that there were over 70 auxin-related genes that were altered by the addition of B. japonicum to the plant. And this also included the several auxin transporters. And these changes were significant, as you can see here. And some of these genes are important for auxin efflux transport. And others, like you can see here, the pins like 7. So, and others were important in the primary root length. So there was that interaction between several of these genes and the pins, and also interaction with them and the ABCB family. And then from here, then we wanted to see to confirm our results, and for that we used several single mutants of the genes that we had seen to be important in lateral root production by B. japonicum. And for each of them, we used 2 alleles. So we have the pins, pin 2, pin 3, pin 7, ABCB4, and ABCB19. And what we found again confirmed our results. We found that pin 3 pin 7 and ABCB19 lacked that increased lateral root effect that is induced by B. japonicum. When they were mutated or they were not functional, then that sudden increase that we saw when we added B. japonicum was missing. While in the case of pin 2, that did not seem to be affected. It did increase. And in the case of the ABCB4, there was also that increase that occurred after we added B. japonicum, indicating that functional pin 3, pin 7, and ABCB19 were required for B. japonicum-induced lateral root growth. Now, the interesting thing for us here was that the pins are redundant in function. They're functionally redundant in several places in the plant. And sometimes you have to have more than one mutation to see an effect. So for example, when pin 3 is mutated, then still you don't see the effect of that mutation because pin 7 plays the role, occupies that place because they have kind of a very similar function. But in our case, we can see that these pin genes individually seem to have a role that they played, in the sense that if when each one of them was mutated, we could still see a phenotype, which was pretty interesting for us. Now, at this stage here, we are seeing that this B. japonicum is having an effect on lateral root formation. But the question is, at what stage in the lateral root formation is having an effect? Is it having an effect? Lateral root formation occurs in several stages. As you can see here, as has been well documented, there are 7 stages. So is B. japonicum just increasing this process, just making sure that this process goes quickly to completion? Or is B. japonicum inducing formation or initiation of other lateral roots? Now, all these processes require auxin. Auxin is a key phytohormone in these processes. And our thinking here was that if B. japonicum just increased the rate at which this process was going towards completion, then the number of lateral roots plus the number of primordia at every other stage, when added together, should be very similar between the control and the B. japonicum inoculated plants. However, if B. japonicum was responsible for initiating lateral root primordia formation, then the number of lateral roots formed and plus the number of root primordia, when added together, would be more in B. japonicum inoculated plants than in the control. So we expected either to have a similar number between control and PGPR inoculated, or a higher number, depending on which on whether B. japonicum was increasing initiation or not. And when we did our study, what we found was that in B. japonicum inoculated plants, in the wild type, when we compare them, we found that the total number of primordia were more in the control than they were in B. japonicum inoculated plants. Here, however, the number of emerged was significantly higher in the B. japonicum inoculated plants compared to the control. And then when we added up the total, we found that B. japonicum had a higher total than the control. There were about over 30 more in B. japonicum inoculated plants, showing us that B. japonicum not only encouraged the emergence of lateral roots, it also helped promote initiation of lateral roots in the plant. Now, for the mutants that we had seen to have an effect, or were for the mutants that we had seen to be affected when whose response to B. japonicum was affected, for those kind of plants, we also found that to be similar, to have similar results. In the case of pin 3 here, we can see that the number of lateral roots were affected when the gene was mutated. It wasn't similar to the control, but it had fewer. And then we had seen pin 7 was involved, and we can see that in the case of pin 7, the number of lateral roots were also fewer here. And then in the case of ABCB19, the number of lateral roots were also fewer in B. japonicum inoculated plants, also confirming the findings we had before. For the genes that we had seen were not really required for lateral root formation, that was ABCB5 4 and pin 2, we see that the change was not that significant in the case of pin 2. And in the case of ABCB4, actually they were higher in B. japonicum inoculated plants. It's a very similar phenotype like the control. And therefore, to summarize the results we have here, we saw that B. japonicum enhanced lateral root initiation and emergence. And we also saw that pin 3, pin 7, and ABCB19 by themselves played a role in acceleration of the growth of lateral roots, and also throughout the formation stages of the lateral root primordia. And also this table also confirmed that what we had observed before, that pin 7, pin 3, and ABCB19 were required for lateral root increase by B. japonicum. And then we wanted to see if B. japonicum also affected distribution of auxin in the plant. And for that, we did some confocal imaging of GFP tagged pin 3, pin 7, and ABCB19. And what we observed was that there was a reduction in the fluorescence emission in pin 3 around the meristem. As you can see, compared to the control, this is a control, and this is the B. japonicum inoculated. You can notice that there is a reduction in the signal around the meristem here. And also in the case of pin 7, we also observed it. And in the case of ABCB19, we also observed that there was a reduction, more reduction than we had expected. And then in the case of ABCB19, we also noticed there was a reduction in the meristematic area. As you can see here in the control, there's this bright signal, which was lacking in the ABCB inoculated plants, indicating that B. japonicum had, of course, was influencing the distribution of pin 7, pin 3, and ABCB19. And then we summarized our data in this table here. And what we included in this schematic diagram was the already known information about the distribution or the location and directionality of the pins and ABCBs. And here shown with the arrows. So pin 2 is responsible for both acropetal and base petal transport of auxin in the roots. This is mainly just at the root tip, and it's shown here in purple. Then ABCB4, shown in black arrows, is responsible for base petal movement. And then ABCB19 and the pin 3 and pin 7 are more located in the central part of the root, and they are more responsible for the acropetal movement towards the tip of the root. And then, however, when we added B. japonicum, we noticed that there was an accumulation of the signal, a bright signal, towards the differentiation zone here, and then also in the meristematic region of the root and towards the root tip, in the meristem towards the root tip. And then in the case of ABCB19, we did notice that also here in the transition zone, we did see that there was an accumulation of the signal and also towards the root tip. Pin 3 and pin 4, pin 3 and pin 7 are also responsible for lateral root movement of auxin towards for other tip. So they move it towards the root, which then, as we can show here in this arrow. And here we have a star here indicating the place where we found most of the root hairs forming in our roots. And here towards on the outside of the root is where we have the bacteria that had surrounded the root. We're demonstrating where the bacteria was located all around the root system. And with that, I would like to thank the people that were involved in this work. Dr. Mercedes Schroeder, who was a postdoc at the time in the lab, was responsible for this work, and she did an excellent job. She was very diligent with the work. And she got some assistance from the grad student at the time, Nathan McLean, and the assistant specialist. This work was funded by some funds, several funds from UCR, and also from USDA, some funds from USDA. And with that, I'd like to thank you very much for your attention, and I'm ready to take your questions.
Thank you, Emma, for a really wonderful talk. Very much enjoyed it. why don't you stop sharing your slides, and we will turn to the question portion. So I wanted to encourage everybody who has a question to please type it into the Q&A, and we'll start there. Okay, Emma, could you stop sharing your screen, please?
Sure.
Thanks. I see lots of thumbs up coming through, so I can tell that it was an interesting talk to many. So let's see. Jose Lopez Bucio has a question. PIN2 mutants have indeed more lateral roots irrespective of bacterization. What could be their role in the response to bacterial auxin?
So in our case here, we do know that PIN2 mutants have a lot of more lateral roots, but we did not see any difference between them and the wild type, and our code 0. Therefore, we did not investigate what their role would have been in because we didn't see a change in phenotype. However, what we did notice about PIN2 was that they were involved in the primary root growth. We did see some interaction there, and therefore, but we did not go into details about it because here we're focusing mainly on the lateral roots.
Right, a very interesting possibility for future work with the primary root. Okay, and we have another question. The increase in lateral root number is due to endogenous cytokinin from the PGPR? That's a question. I know you measured auxin, but maybe you could speculate about whether the PGPR, whether the Bradyrhizobium might be producing cytokinin as well, which could also influence.
Yeah, thanks for the question. That is also a possibility, but we chose to focus on auxin. But cytokinin also does play a role in the root architecture. And the reason we chose to focus on auxin is because auxin and cytokinin have this antagonistic role that they play. And we thought that we'd be pulled in two directions and we not have a focus. So in future, we'll focus on cytokinin, and for those people that are more cytokinin lovers than auxin, but also auxin gave us this very clear path. So we thought if we focus on auxin, then we could focus on cytokinin at a later time. But thank you for your question. That's true. Cytokinin does play a role in lateral root formation.
And of course, we know some Bradyrhizobia produce abscisic acid, something that I'm interested in. So lots of different possibilities. These bacteria have clearly evolved to manipulate their plant hosts by producing plant hormones. Fascinating story. Okay, William Teal asks, do your data indicate more PIN3, PIN7 transcript after inoculation, and yet a reduction in PIN GFP signal? Does this imply a post-transcriptional layer of regulation?
What I would say is that let's not mistake an increase in expression with distribution, because we are only focusing on the tip of the root. So there could be a possibility that this is just due to distribution. It focused it where it needed to be for lateral root formation, but we didn't go into a lot of details in that. So there are all kinds of possibilities. And I'd be wrong to tell you what I think it is, because I could be totally off. But our data mainly showed effects on expression and distribution. But where it is taken to, we don't know. So yeah, thank you for your question, but it's something that we will not be able to answer right now.
Mm-hmm. But yes, there's this whole, it's, you know, as we move from an era where we grind up the whole root and measure things, and now we're looking at a much more cell by cell or tissue layer level, we're starting to see interesting things that we could not see before. Okay, Avinash Sharma asks, as you said that the bacteria secretes auxin into the medium, which induces lateral root initiation, my question is, how important is the presence of the bacteria itself? Yes, and you kind of hinted at that in one of your figures, that the bacteria is not the same as just adding auxin. Can we treat the plants with medium alone after removing the bacteria and expect the same results?
Yeah, we did have one of our graphs, I think, I don't remember what slide it was, where I showed the addition of auxin influx inhibitors. And we removed the bacteria and just used the bacterial culture itself. And we showed that the results were comparable. So the presence of the bacterium itself was not necessarily required, I would say, for some time, because the bacteria is a living organism and it's always producing auxin. It's producing auxin over time. So you'll come to, if you just use the culture without the bacteria, you will exhaust the resources and then you will need the auxin again. The other thing is that just using the, removing the bacteria and just using the culture filtrate, initially you have the effects as we had, but also I'm wondering about the feedback mechanism, because bacteria are living organism, and if there's too much auxin, they most likely will stop producing. And then if the auxin is needed again, then they start producing it again. So you will, you can use bacteria in the media, but you're going to, no, you can remove bacteria and just use a medium, but you're going to lose that effect of the living organism that triggers more than just auxin. There's more than just auxin being produced by bacteria, by the bacteria, if you ask me.
Definitely. I also wonder about the feedback from the plant onto auxin production, you know, that what's stimulating them to produce it, and presumably there'd be some feedback there at some point to fine tune the.
Yeah. And I'm also like, I showed a graph where we had addition of NAA and there was this spike in lateral reproduction, just quick and very fast, and leveled out at the end. And the bacteria were slow because we inoculate a very small quantity of bacteria and then they build up over time. But then the auxin that you added, the exogenous auxin that's added, at some point is going to deplete. But like I said, the bacteria will continue having this interaction, and every new roots that are formed are going to be having an interaction with the bacteria. And therefore, there is more to the bacteria. We know all the other factors that PGPR lead to or trigger in the plants. So that's going to be missing if you just add the exogenous source of auxin rather than just having the bacteria itself.
Definitely. Okay, we have another question from, I'm sorry, I'm going to try the name, Owen King. Did you observe modification of JA-related genes in your RNA-seq data set?
There was probably some, but we only, like the ones we have for this paper were only selected for auxin. But in another paper, we did see another paper that's coming up in BMC. We did see a change in JA-related genes. So, but I don't have the data here to show you. But we did see some changes in JA. You're correct.
Interesting. So much feedback back and forth. It's really a network of hormone signaling. I always think of it that when you tug one edge of the net and it affects everything. Okay, Pascal Raté asks, is there a type 3 secretion system in this bacterium? And can the system interfere with PIN gene regulation?
We haven't confirmed, but bacteria are known to have a type 3 secretion system. So we haven't confirmed that's something that we could do in the future. But thank you for the question. Just gives us more to do. Yes, we'll check on that and maybe tell you in maybe sometime soon.
Yeah, that's an interesting question. We're starting to see more and more effectors coming from at least some of the rhizobia, certainly from some Bradyrhizobia. It'll be very interesting to see how that's one of the tools that's used in this interaction or not.
Yeah.
Okay, Stephen Pullman says, asks, your DR5 GUS data shown auxin signaling all over the place. How does this result in local auxin gradients to activate lateral root primordia outgrowth?
That is an interesting question because we would have expected the bacteria to be more organized and just put the auxin where it's required. But notice this is a very young seedling we're dealing with. This is a 4-day-old seedling. Therefore, when you add the bacteria, I think initially it's going to be all over, and then it's going to be harnessed by some auxin-responsive genes like GS3, and which will conjugate whatever the plant doesn't need, and then it's going to move limit them to being all over the place, because he's right that having auxin all over the place will interfere with the gradient. And but we did not do this study. We did not follow up with older roots, which could be interesting to look at and see the distribution again in older roots. But we only focus on younger roots very, very early in the development and soon after adding the bacteria. So there's a possibility of it being conjugated later on.
One other thought to keep in mind is always with Gus fusions is that the enzyme is so stable that when you stain for it, you know, that enzyme could have been produced 24 hours ago and then is no longer being transcribed. But of course, you have the enzyme there happy to do its periodic enzymatic reactions. You always have that little background with Gus that you're seeing sort of the sum of events rather than a momentary thing. But it's still, I agree that I suspect it's high and then goes down, and you're just seeing the relic of the high. Okay, Sunil K asks, very interesting observations, Emma. I am fascinated. Why don't you look into the bacterial factors responsible for such an increase? Are you doing it now? So I think what this question is getting at is the bacterial factors responsible for maybe the production of auxin.
Yeah, that's an interesting question. We did think of doing it at that time, but we were trying to balance and seeing which one we were more excited about. But there definitely there's a need to look at the bacterial factors, and we will need to generate some bacterial mutants and see which of the factors in the bacteria are responsible for it. But at the time, we were more curious about the plant. But now, as we get more data about the plant, then we might want to fall back to the bacteria and see what it is doing and how it's causing all this. So there's so much we can do in this study, and I'm excited at how much more we can still dig out of it.
Yes, definitely. And we have Brian Push asks a question, sort of following up on that, really. Do you find evidence that B. japonicum entered the roots as one means by which the roots showed increased auxin? And I guess you do have that difference between where you showed the graph that had the effect of the culture filtrate versus the rhizobia. And so this is really, I guess, kind of asking, what do you think the Bradyrhizobia are doing? Do you think that they do enter the roots?
No, right now we don't think so. The reason we don't think so is we have tried to do several kind of studies whereby we wash the roots, we do the bacterial count, we grind the roots, we do the bacterial count. So we never see it increasing with getting tissue out of the roots, but we could be wrong. So that's why we haven't, with confidence, published out there and said it does not enter the roots. And then when we have tried to do some confocal imaging, and we have found that that even within the layers, we could see bacteria, and we were not sure why. We were not sure if it was because of the way we were imaging our plants, or it was because of the way that our slicing was not good. So we didn't have the confidence to say it's not there or it is there. So we have done a few studies, tried to see if it's really going in. And for right now, we can say we don't think it's going in. Is there a possibility that it's going to the roots? Yes, there's that possibility. But we don't see any nodules being formed. But it could go in and be an endophyte. But we don't want to comment on that right now. We cannot say yes or no. We need to have more conclusive data before we can comment on that.
Yes, that's a really good question, an interesting idea. And it is so hard. If you see it, you can say yes, but if you don't, it's really hard to know, because it could be that just a small number of bacteria are having a big effect.
Yeah.
So many questions remain for the future there.
Yeah.
Okay, Avinash Sharma says, Also, my next question is, at what stage do you treat Arabidopsis in order to get more seeds and more pods? Do you have to include the bacteria in the medium throughout the time of plant growth?
Well, we have done this work both in the soil and on our growth medium. When we inoculate in the soil, we let the plants germinate And after they have germinated, we add a small portion of the bacteria at the root tip of the plant, and that's it. We don't add any more times. We just add one more time, because we are this school of thought that if the bacteria is to do some work, it has to be self-sustaining. So we don't have to come in and keep on inoculating. It has to be able to build up its own population. So for all those experiments, our inoculation was only done once. However, if somebody is doing it out there in the field, if they felt like doing more than once, maybe that if those need to do more, then I think they should, they could, if they felt that they could get better results with it. So it's not a rule. It's not something we're going to be saying that, okay, you only need to do it once. But in our experiments, we've only done it once to retain uniformity. And we have seen all these different expressions.
Right. I mean, it was originally isolated from the rhizosphere, right? So it should be able to survive and grow under those conditions. Good point. So Raviraj Banakar says, exciting work and thanks for the presentation. Does increased lateral roots influence nitrogen uptake and ultimately the phenotype?
This here I'd have to speculate, because I would say that increase definitely in number of lateral roots is something that's of benefit to the plants. It will increase uptake of all minerals that are required by the plants. Anything that's able to uptake using the root system, it's going to be able to uptake more efficiently if it has a robust root system. It's not only nitrogen, it's also phosphorus, the elements like iron, calcium, whatever else the plant needs, it's going to be able to uptake. The other thing is that it increases the surface area. When you think of lateral roots increase, this increase in surface area. So that whatever the plants need to interact with the soil, it has a larger surface for that interaction. So I would say yes, it does increase nitrogen uptake, and then also then helps the plant to grow bigger because it's providing the plant with more nutrient, more water absorption. If water is a limiting factor, that would also be a good thing for the plant to have a more robust root system. And anyway, whenever you go somewhere and you find a very big tree, and somebody tells you to dig up the root system, you'd always think, I'm thinking it has a very big root system because it has to feed the whole of this above ground system. So the better the root system, the better you are off with the plant. It's going to be a bigger plant for sure.
Right. So now we have a question from Alafia Motiwala, who asks, what is the minimal quantity CFUs of Bradyrhizobium japonicum used to see the effects of lateral root growth? And also asks a second question, does it form a biofilm layer? Is it maybe having an effect? Another aspect of its growth promoting effect could be by interacting with other microbes in the soil, or as they point out, preventing, perhaps the biofilm prevents other bacteria in the soil from interacting with the roots.
Okay, that's a loaded question, and thank you for asking. So you're asking first, I'll do it stepwise. You're asking the minimum. We have done a series of dilutions, and we see effects even with very little added into it. I think even with 10 to the power of 2 added to it, CFUs. But the significant results. So if you're looking for significance, then we go to about 10 to the power of, starting from 10 to the power of 6. That's when we see significant effects. And if you want significant results quicker, then you have to increase the population of the bacteria. But with 10 to the power of 8, we can see a significance starting from 2 days, moving on. But we do see differences. Like, I don't want to say it's only that concentration. We do see differences even to the 10 to the power of 2. It's just that they were not significant at the time we measured. But given time, based on you can see the way we did our work, but given time, 10 to the power of 2 would also multiply and give you the results you're expecting. But it depends on when you want the results. So any quantity of the bacteria, as long as it's able to multiply in your environment, it's going to produce some significant results. But the higher the bacterial concentration, the quicker you see the effects. And then he had a question about the biofilm. If you don't mind, would you read that again, Ginny?
Yes, that was asking about, does it form a biofilm This is back to, I'm assuming biofilm on the surface, since you said it probably isn't in the root, or may not be in the root. So biofilm on the surface. And does this prevent other bacteria in the soil from interacting with the roots?
I'll start with the last part where you're asking, does it prevent other bacteria from interacting with the roots? I would say probably not, because I don't think it's the only bacteria that you would find in the soil interacting with the plant roots. So we haven't done that study, but I would say for sure, I don't think it would stop other bacteria from interacting with the roots, because that would mean that it has a monopoly with the plant, which is very unlikely. There's this beneficial interaction between the plants and the different bacteria species, and also the interaction between the bacteria and other organisms in the soil. So this microbiome that exists in every root rhizosphere. So I wouldn't think that it would stop the roots from colonization. It would stop colonization of roots by other organisms. However, we haven't done that work to see if it's going to limit other bacteria from entering into the root or having an interaction with the root. And also, we haven't done a study with the biofilm yet. And thank you for bringing it up. I think this presenting just opened up how much we haven't done, okay, and how much more we need to do. So we haven't done the biofilm studies, and we will for sure look into that in the near future. Thank you.
So many exciting questions. So many exciting questions.
Yeah.
I'm glad that it's giving you a lot of ideas as well.
Yeah.
As clearly the listeners. And Sarah Canavo asks, sort of continuing on that direction, could you tell us more about the biology of the bacterium and what kind of symbiosis it forms with the plant? And so I think you could answer that in different ways.
Yeah. I don't think we have done, we have like broken down every possible interaction or studied the interaction completely with the plant. But we are thinking that the plant root exudates provide carbon as they do for other plants. If you have studied plants, you find that they about between 25 and 40% of the photosynthetes are released as root exudates in the rhizosphere. And these are to feed the microbiome that is in the root. It's in the rhizosphere of that plant, and therefore it's going to attract all kinds of bacteria. It would attract both pathogenic and beneficial bacteria, and also attracts fungi like mycorrhizal fungi to the rhizosphere of the plant. Now, we have not studied exactly how the plant is releasing root exudates to attract this bacteria. We haven't done that extensive study, but that's also another possibility. But we think the bacteria responds to the plant root exudates, which provide carbon, and then the bacteria is producing nitrogen, it's producing IA, which is auxin, a form of auxin, which the plant takes up. And then plants also in the root exudates release tryptophan. So if tryptophan is then being released in the root exudates, then the bacteria produces more IA, which the plant takes up. And then there's this continuous beneficial interaction between the bacteria and the plant. I think that question had more than just one part.
No, I think that's a good try on it, a good take on it. It makes me now wonder what would happen if you took the exudates, just sort of took a root wash basically and put it on the bacterium.
Yeah.
What's that going to change about what they produce in their exudates? How's that going to affect auxin production? How does it affect gene expression at their end? So that whole back and forth.
Yeah. I haven't done this study with the bacteria. I've done it with mycorrhizal fungi, and we could see different effects at that time. I was doing it when I was a postdoc. So there's that possibility. And also, like I said, this just showing us the many more studies we could do. And which should also be exciting and provide better understanding of the interaction between the bacteria and the plant.
So many questions to ask as you are doing your confocal studies, looking at, trying to see whether the bacterium is actually inside the root or not. I'm also wondering whether you're noticing patterns of its attachment on the root. Like, do you see more of it in areas of lateral root primordia or lateral root emergence, more of it near root tips? that will be a fun thing to look at in the
Yeah.
future.
Yeah, the thing that complicated that is because we put the root in there, but we have to put a solution. So then we dislodge the bacteria. Then when minute we put the root, the cover sleeve, and then the bacteria starts swimming all over the place. So then we don't know where they were originally, whether they were at the tip or they were on the in the differentiation zone. So then we decided to relax on that because we were not sure where they.
Perhaps you could inoculate them and then 24 hours later do a slight wash and then put it on, and only the ones that were attached would stay.
Yeah, I think that's
and then
that's
you might be able to
possibility.
answer that.
Yeah.
Interesting question. Great ideas everybody's coming up with. Lots of great discussion here. Amit Kesharwani asks, cytokinin dehydrogenase also helps increase root development in plants. Have you evaluated the combined effect of cytokinin oxidase and auxins during your study?
No, not yet. Thank you for the idea. We are going to consider doing cytokinin. There seems to be a lot of cytokinin people out there. We will see if we can do that study between cytokinin and auxin.
Lots of interest in cytokinins. So Subramanian asks, have you thought of the legume symbiosis angle of auxin production? Bradyrhizobium japonicum is a nodulating bacteria for soybean.
Yeah, we do know that Bradyrhizobium japonicum, USDA 110, induces nodulation, but this is not the same species. So this one did not induce nodulation. And we did try to see if it was going to induce nodulation. So we did grow it together with a few legumes there, and we did not see nodulation. So it's a different bacterium. This one is not inside the root, not inducing nodulation. So in that sense, then we haven't studied that symbiotic interaction as you would find it in the case of soybean and Bradyrhizobium japonicum. So we don't have that kind of interaction here.
Yes, that brings me back to when I first started studying about, as a student, reading about Pseudomonas and never noting the strains, and suddenly realizing at the end that was why I couldn't make sense of everything, that different strains had different functions. So I think that's another thing we're seeing here. Yes, Bradyrhizobium japonicum, but it's not the same. Bacteria are amazing. Okay, so we have another question here. How would you explain the increase of lateral roots in pin 2, but a decrease in pin 2 and pin 7?
Say that again.
I'm not quite sure either. How would you explain the increase of lateral roots in pin 2, but a decrease in pin 2? But I'm not sure.
Maybe they mean pin 3 and pin 7?
Maybe. Why don't you answer it that way?
Yeah, then okay, I'll take it that you're asking how would explain an increase in lateral roots in pin 2, but a decrease in pin 3 and pin 7. So all these pins have different roles in the roots. And in pin 2, even with a mutation, it did not affect lateral root formation, because in our case, we would say all the other pins were functional. And therefore, like I said, there's a redundancy in their functions. And also maybe pin 2 is not really required for in B. japonicum induced lateral root increase. But in the case of pin 7 and pin 3, we did notice that when they were mutated, then the lateral roots, the induced lateral roots by B. japonicum did not occur. But they did have the same number of lateral roots as a control. So, but the induced increase was not there. And that's why we were saying that a function of pin 7 and pin 3 individually are required for lateral root production. I also did mention that the pins have a redundant roles in the plant, in that sometimes when you mutate one, you're not likely to see a phenotype. And in most cases, you have double mutation or triple mutations for there to be a phenotype. But in our case, we did notice a phenotype even with one mutation, indicating that each of the pins individually did play a role in the bacterial interaction in inducing lateral root formation. So I would say that we were happy to see those results, because otherwise we would have had to have several mutations, like double mutants or triple mutants, to see an effect. So I think that's the explanation we have here. Each of the pins, pin 3 and pin 7, play individually a role in lateral root induction by B. japonicum, but pin 2 does not. And that's why we did not see any difference. I hope I'm answering your question.
The next question is similar, but with respect to primary root. So I think in the interest of time, we're going to pass on that, although of course it is an interesting question, primary root growth. And we're going to go to Dr. Mohammed Shahzad, who is asking, do you think this Bradyrhizobium strain might have TAL effectors? And you had said previously you have not looked at effectors yet, but they're wondering whether that could target Auxin genes directly in the root.
Yeah, there's a possibility. There's a possibility of that, but we haven't studied it. So I would not want to mislead you and say yes or no, but it's something that we are thinking we should look at. There are a lot of people asking for effectors on type 3 secretions. Then we will go in that direction soon.
Yeah, there's a very nice story about Bradyrhizobium effectors affecting host range in, I guess, soybean recently. So that's intriguing. Okay, and Sridhar Ranganathan asks, does this effect of the Bradyrhizobium inoculation last for succeeding generations of the plants? So I guess that means if you took those seeds from the saliques and planted them in fresh soil that you had not inoculated.
Interestingly, yes, but we didn't study it. Interestingly, we did see that. Yeah, but we did see that, but we did not study it because we were not even finished with the first generation. And then we're going to start hopping to the second one. But we did see that increase, that the seeds were better, healthier. That's what the way our interpretation was. They were healthier, they're more robust. And we did not have an explanation for that epigenetic effect, but we observed it. We did observe that it did last more than a single generation.
Those transgenerational effects are so fascinating, and so many of them are just at that level of anecdotal observation at this point. But yeah, you can only follow up so many things, but you've raised a lot of exciting ideas and directions to our audience. And I think we are going to, well, why don't we stop with, well, I'll just read Jose Lopez Bucio's comment. Maybe it is not really auxin itself that is the lateral root inducing agent, but the repression of primary growth by another factor, because that reduces apical dominance. Yes, if you reduce primary root growth, it stimulates lateral root growth. So interesting idea there. Avinash Sharma then says, out of curiosity, this is a good final question. What does the Bradyrhizobium japonicum get as a reward or benefit by secreting auxin in the medium? Why would bacteria be making and secreting auxin for plants? And I think the answer is really that it's not secreting the auxin for the plants, it's secreting the auxin for the bacterium. So if you want to go that direction.
Yeah, I think there's that, yeah, in the medium, I think it might not secrete too much if there's the absence of the plant, because by itself, it doesn't need the auxin, or maybe it does, I don't know. But, you know, I think human beings are naturally selfish. So we're just looking at the yield we're going to get, the plant biomass we're going to get. So we're not thinking of why the bacteria would do that. But there's a possibility the bacteria by itself might benefit from it, but we don't know. But from our point of view, we're mainly looking at it with its interaction with the plant and how the plant is going to benefit, and the benefits we are going to get with increased biomass and increased yield. But I don't see the need for excessive production of auxin in growth medium by the bacteria. I don't see there is that, unless, like in our case, when we supplied tryptophan, I think we tricked it to produce more. So
I think when I look at it, I think one thing the Bradyrhizobium is doing is basically generating more habitat for itself, right? If you live on the roots of these plants and you can induce the host to make a lot more roots, you have just made, you know, presumably they're living off the exudate. You've just made a much larger root that's producing more exudates. So I think that's what they're really doing, is increasing their habitat and at some level their food source. I don't think they're doing it for the plant, but that's handy for us. So, okay, I think we're going to stop there. This has been a terrific seminar. Thank you so much, Emma, for presenting your work to us. It's so exciting to have our readers be able to ask you questions directly on all sorts of topics, and that's been a lot of fun. I really appreciate your stamina and answering more than 30 minutes of questions. So we do like to go as long as people have questions, because that's really one of the points here, is inclusion and giving people a chance to be in the room and ask the questions. And while I say that, I want to remind everyone that if you missed it, or you missed part of it, or you have friends who would like to watch it, please check back where you registered, and there will be the link to the recording, which we hope to post soon, maybe even later today. We'll see. So thanks so much for attending, and thanks so much, Emma. And also thank you. We had one volunteer to assist with the transcript, Drishti Sarkar. So thanks for that. Okay, bye everyone. See you next time.
Yeah, thank you everybody. Bye.